Right, so here endeth the lesson, and I'll start back on microbiology. And so 2 lectures left from me. So today we'll be talking about the human microbiome, that is all of the commensal bacteria that live in or on the human body without causing us any problems. And tomorrow, I guess probably tomorrow will be bacterial pathogens. Okay, so the human microbiome. Now there's a kind of frequently cited number, which is that there are 100 times more bacterial cells than human cells in our bodies. But apparently this is not very accurate. So it's been, this number's been revisited relatively recently, because looking at where this figure came from, 100 times more bacterial cells than human cells, it turned out that the numbers were from papers in the 1970s, where they just gave a very, very rough estimation. And then you've got this kind of phenomenon where somebody's back of the envelope calculation is repeated and seems to be taken as the truth. So this was a kind of look at this number again just last year to try and give a better estimate, or rather test whether it's true or not. So apparently the number of human cells in our body for a 60 kilogram adult will be about 3 times 10 to the 13. But surprisingly, most of these are erythrocytes in terms of, numbers, not in terms of the mass contribution to the body, but in terms of numbers of cells. Most of the cells in our body are erythrocytes. So if you just limit the consideration to nucleated cells, then we have a 5 times 10 to the 12. Now, in bacterial numbers, from all the sites that have been sampled and counted the total number of bacteria, the estimate is that in the colon there's about 4 times 10 to the 13 bacteria. And on the rest of the body, mouth, genital mucosa, skin, then you'll have 1 times 10 to the 12, or maybe a bit more. So that means that the overall ratio of bacterial to human nucleated cells is about 8 to 1. It was not 100 to 1. In fact, about an order of magnitude less than that. But nevertheless, it is true that the number of bacterial cells on our body outnumbers the number of our own cells. So really the idea of studying the human microbiome is the realization that these bacteria are always with us, where the human body is colonized by bacteria soon after birth. And they are commensals, they're symbiotic, so they contribute functions to the tissues which they colonize. So we want to understand those functions. Now, how are we going to go about this to study these bacteria and figure out what they're doing? Now, it's not totally a new subject. So people have been looking at microbes on the human body for, I don't know, maybe 100 years. And classically, what you would do was take a sample from some site on the human body, dilute it out, and then spread it on agar plates, and then try and identify all the colonies by biochemical or serological means. Something like what you'll be doing in the lab classes. So for example, you could start with, somebody's feces, make a kind of, mix it up, make some dilutions, and spread them on selective plates for gram-negatives and gram-positive bacteria. Put the plates in culture in the presence or in the absence of oxygen, and then you'll have an idea of how many aerobic and how many anaerobic gram-positives and gram-negatives you have per gram of feces. And then in amongst those 4 categories, then you would start picking out 100 colonies and identifying them. And that will give you an idea of, what the fecal microbiome is like. Now, the drawback of this type of approach is kind of twofold. One of them is that it's very labor intensive. So you can imagine, for gram-positive, gram-negative aerobes and anaerobes, if you just pick 100 per category, then you have to perform 400 identifications with classical microbiological techniques. So you have to set up a lot of cultures, and it's going to take time. So you could maybe do it for 2 or 3 or 4 people or something like that. But if you want to have a population level data about the human microbiome in different people and whether it correlates with disease or not, then really it's just going to be too much work. Okay? And of course, because it's so labor intensive, you can get information only about bacteria that are present, at least that make up at least 1 or 2% of the total bacterial flora. So if you've got 1 or 2 species that are very, very predominant and a whole bunch of other species that are rarer, you might not be getting accurate information on all of these rare species. Just because you don't have the time to study more than a couple of 100 colonies at once from a sample. So that's one drawback. The other one is that, even if you take care to try and make your culture conditions as all-inclusive as possible, you're never going to be sure that there aren't some bacteria in the sample that don't really grow very well on the conditions that you choose. So you have a selection bias because of the culture. Now, people were aware of these these limitations, and for the last, 25 or so years, there's been an application of molecular techniques to study the human microbiome. So the first one is using genes coding for the small ribosomal subunit RNA, 16S RNA. So if you remember, there are conserved regions in this sequence and there are variable regions. So if you choose primers that hybridize to the conserved regions, you can amplify up a PCR product that has, that represents the 16S RNA genes from all of the bacteria that are in your sample. And then you can clone out this PCR product and sequence a bunch of molecular clones. Just with standard Sanger sequencing. Now that gets out of the culture bias because you're not you will be able to detect microbes that don't grow very well in the culture. And it's a little bit better in terms of throughput because it's easier to get a clone that carries one of these molecular sequences and sequence the DNA. This is quite easy. It's easier to do than to run a whole bunch of microbiological tests. But still, you're talking about, you can do a few hundred samples, a couple of hundred samples, a couple of hundred clones per biological sample. And what has really changed this field is, within the last 10 years, the development of next-generation sequencing technologies that allow you to sequence in parallel. You can get something like, 100,000 distinct short sequences from a single sample. And you can do this either with the same kind of ID. You get a PCR product that's amplified up the 16S rRNA genes, and then put this PCR product through a high throughput DNA sequencing machine. And instead of getting information on 100 or 200 clones, you get like 1,000 times more information. So this is really much more powerful. And with kind of multiplexing, you can add sequence tags on either side, so you can decide, I want to get data from 10,000 clones from 10 samples, and that will be one run in your sequencing machine. So in terms of experimental power, this is really what allows people to study a whole bunch of samples from different people and get a huge amount of data. The next step up is to not use the PCR step, but just extract genomic DNA from the sample and sequence everything. So you'll have a bunch of short sequences from the DNA from the sample, and then bioinformatically they'll be joined together where they overlap to reconstitute the whole genomes of all the bacteria that are in the sample. So this will give you more information about, the metabolic properties of the bacteria that are in there, whether you have any pathogens, whether you have virulence genes or not. And both of these techniques are very, very intensive in terms of computing power. So experimentally, there's less work at the bench to do, way much less than back here with the classical microbiological techniques, but you have a lot more bioinformatics that's involved in this kind of microbiological research. So one of the big projects that was launched with this technology was the Human Microbiome Project. They published data in 2012, and they processed almost 5,000 samples from 242 healthy individuals. And they were describing the microbiome present at 15 or 18 different anatomical sites. And the summary is here. So here are the different anatomical sites, okay, skin, mouth, feces, and vaginal mucosa. So this is why there are 15 in sub-subjects, so okay, 15 sites in the men, 18 anatomical sites in the women who were enrolled. And here are the different bacterial genera that were mostly found in these sites. Okay, so the size of the circle here is represents how prevalent these different bacterial genera were at the different sites. So what can you see from this figure? Well, one of the things you can see is that the different sites have got a different microbiome. You don't have the same bacterial genus that is prevalent all over the human body. Okay, so in the mouth, you've got Streptococcus, Haemophilus, Prevotella, and Veillonella. Pretty much, whereas, in the feces, you've got a lot of Bacteroides. Skin is also different. Okay, well, so different anatomical sites have got different microbiota. The other thing is that within a different locations within the same tissue, there's not that different. Okay, look at all this stuff in the mouth. They had people picking out swabs from all kind of different places in the mouth, and it must have been very disappointing when they got the results here. Okay, like they did 1, 2, 3, 4, 5, like 6 sets of data, which are pretty much all the same. So that's 6 times more work just to show that you're getting the same kind of stuff. The exception here is in the plaque. Okay, so you've got a lot of Corynebacterium in the plaque. So you've got 2 different environments within the mouth. You've got the plaque, and then you've got all over the surface of the mucosa in the mouth. And you have a similar kind of thing in the skin with similar profiles pretty much wherever the sample was taken. Not exactly the same, okay? So around the nose, this is a little bit different here. The antecubital fossa, this is here, where your elbow is bending. And that's from behind the ear. So some differences in the body side. And then a couple of So here the microbiome is quite mixed, skin and mouth, but in the feces and vagina mucosa, it seems like you've got one big species of bacteria that dominates the microbiome, and then other bunches of rare stuff. So what I'm going to do now is go through these different sites and have a look, see what kind of bacteria are there, and try and explain what their functions are. So we'll start with the mouth. Well, here, not really the functions, but how these different bacteria interact. Okay, so this is a paper from data from a paper published last year. And they looked at the human oral microbiome database. Yes, there is such a thing. And apparently there are 700 species of bacteria that have been found and identified in the human mouth. Not all in the same person, okay? So this is an aggregate of all the studies that have been done. But in an individual, you'll probably have, a couple of hundred different species. And the authors of this study, they're particularly interested in the dental plaque microbiome. And within dental plaque, they found that there are 13 genera that are abundant overall. And of those, so that's these 13 that are represented here. So 2 different locations within the plaque. And each bar here represents a different genus of bacteria. And the size of the bar is the prevalence of that bacterium. So you've got, as I said before, a lot of Corynebacterium, Fusobacterium, and Streptococcus. Those are probably the big 3. Okay, and then you have a bunch of others. Some of them, they say, of these 13 are rather rarer, Laurotropia, and maybe these Actinomyces, and Rothia. So I haven't given any information about these 2. But so you have an idea of what these things are. This is whether they're gram-positive, gram-negative, aerobic, anaerobic. Okay, so in the lab classes, when you are invited to inoculate an agar plate with some sample, you can use saliva if you like. Okay, so you should use this kind of information to give you an idea of what kind of bacteria might grow. Okay. So that's all the bacteria that are in plaque. And the next step that they wanted to do was to try and figure out how these different species interact with each other. So what they did was they took pieces of plaque from somebody's mouth, embedded them in epoxy resin, and then cut thin sections, and then hybridize the sections with a fluorescent probe, which is going to stain specific sequences of RNA that are characteristic for each genus. So what they've got here is purple is a Corynebacterium probe, green is a Streptococcus. So they found these kind of things that they describe as being like a corn cob or a cauliflower. So you've got filaments of Corynebacterium down at the base, and then on the crown a lot of Streptococcus. And if you look further, and also there's a kind of gap between them. So you've got the Corynebacterium, then a gap, and then the Streptococcus. So then they went ahead and did a similar kind of experiment with a whole bunch of probes for all of these 8 genera, which were the most prevalent. And so what they found was the same kind of thing. You've got Corynebacterium at the base. Then you've got a kind of intermediate section here with a lot of Fusobacterium, a lot of Leptotrichia. And then in amongst the Streptococcus, you've got Haemophilus, you've got Neisseria, maybe a bit of Porphyromonas, this dark blue. And here Capnocytophaga as well, kind of in the intermediate section. So I found this very interesting because what it's telling you is that your dental plaque is not just a kind of horrible mixture of bacteria. Well, it is, but it's an organized horrible mixture of bacteria. So you've got Corynebacterium at the base, and these will be fixed at the base of your tooth where it inserts into the gum. And this is an anoxic environment, okay, because it's stuck in between the tooth and the gum. So then these Corynebacterium, they grow out, they form filaments, and these filaments will structure the whole of the bacterial community. So afterwards, so at the base this is anoxic, in the intermediate stage, very poor in oxygen, and so this is where you'll get the anaerobes living, okay? Corynebacterium, Fusobacterium, and the Leptotrichia will be in the basal and the intermediate layers. And then at the top, that's where you have aerobes, which start to colonize the Corynebacterium. So that's where you've got Neisseria, Streptococcus. So this is an organized microbial community that will be very stable, and it will grow back in the same way. So when you go to the dentist and the dentist removes all your plaque, then they won't be removing the Corynebacterium that are right at the base. So these Corynebacterium will grow back, and that's why your plaque will reform, and it will have the same kind of microbial characteristics. Not necessarily a harmful thing, okay? So if you don't eat too much sugar, this microbial community will not be damaging to your teeth, okay? Aesthetically, maybe you don't like it, so you go to the dentist and get your teeth polished. But it's not necessarily harmful, unless you eat a load of sugar, in which case the Streptococcus will love it, start fermenting, and produce a load of acid, which will attack your tooth enamel. Okay, so that's the mouth. Let's take a trip down the alimentary canal. So, actually, I might close this one as well. So as you go from the mouth into the stomach, not very many live bacteria there because the acid is going to kill them all. In the duodenum, same kind of thing, very, very few bacteria. From the jejunum medium in the small intestine, you've got a reasonable number of bacteria living in there. It's not huge amounts, but it's not a sterile environment by any means. And really, you have most of the bacteria in your body in the colon, where you've got 10,000 to 1,000,000 times more bacteria than in the small intestine. So there have been several studies of the gut microbiome, mostly done on fecal samples. And fecal samples are representative of the contents of the colon, but not necessarily representative of what you have in the small intestine. But of course, it's much more difficult to perform a study on these bacteria because it's an invasive procedure to go in and sample them. And it's not so easy to get volunteers who are going to undergo an endoscopy, especially if you're talking about healthy people who don't need this procedure for their medical care. So most of work has been on fecal bacteria, and that's what I'm going to talk about now. So here's data from a paper from 2011, where I can't remember how many people, but anyway, analyzed the fecal microbiome from a group of healthy people. And it's characterized at the class level of classification. Okay, so it's not genera, it's not families, it's not even orders, it's really high-level classification, classes of bacteria. And what you see is that the gut microbiome is dominated by these gram-positive and gram-negative anaerobes. So these firmicutes will be something like Faecalibacterium, Bacteroidetes, Bacteroides, and other genera in that class that you've probably never heard about. Because when people talk about gut bacteria, you're probably going to think about Escherichia coli and other enterobacteria. But in fact, this is a very small part of the gut microbiome. It's down here, this one, proteobacteria. So we're talking about, less than 1% of total gut microorganisms. And the reason why people know about these ones is because they are facultative anaerobes. So you spread them on an agar plate, put them in the incubator in the presence of oxygen, and they will still grow. All of these guys, they won't grow. So we know a lot about these bacteria because it's easier to grow them. Okay, so that's at the class level of classification. You have a diverse microbiome. How about species? So this is data from another paper where they looked at 3 different populations of healthy people with different ages. People in Africa, South America, and in North America. And there's quite a bit of different data on this graph. So here, right, on the x-axis, this is the age of the person who was studied. Here on the y-axis, this is the number of different OTUs. That means operational taxonomic units, so basically species. Okay. A number of different species in the sample. So from birth to the age of 3 years, this is when you acquire your gut microbiome. Diversity is really increasing up to 3 years, and then it's pretty much staying stable for the rest of life. In terms of the number of different bacterial species that you have, and in an adult person, you're talking about somewhere between 1,000 to, I don't know, maybe 1,600, 1,800 different bacterial species in your colon. The other thing they found is that there are population differences. Okay, so globally, the colon microbiome was, or fecal microbiome was less diverse in Americans than in South Americans or Africans. So this, in the paper, they put it down to diet. Okay, so this is going to be a more protein and fat-rich diet, and here in Malawi, this is going to be more carbohydrate-based diet with a lot of dietary fiber. And this seems to be related to a more diverse gut microbiome. So, got a load of bacteria in there. What are they doing? Because most of the time they're not doing anything harmful anyway. One of the things is synthesizing vitamins that we need. So it's possible to produce mice that are entirely germ-free. Okay, they can be delivered by cesarean section into an entirely sterile environment and fed with sterile milk from the day they are born. So these mice, which are called germ-free, do not have any gut microflora. But they won't grow very well unless you give them vitamin supplements. So this is a proof that the gut microbes are necessary for the synthesis of some vitamins. Another function that everybody knows about is they're involved in digesting fiber. And this is important for nutrition because some types of carbohydrate are very, very difficult to digest. And the anaerobes that can degrade this type of polysaccharide can produce lactate, acetate, short-chain fatty acids that we can absorb, and we can use them for making fat or respire them to produce energy. So it's a way for us to extract nutritional content from food that otherwise we couldn't digest. So those are kind of the two obvious functions of the gut microbiome. And I'm going to talk about these three in a little bit more detail. Development of the gut immune system, resistance to bacterial pathogens, and the regulation of your and my physiology. Okay, so for the immune system, for the development and regulation of the gut immune system, it's very important that you don't have an immune response against food that you eat or against the commensal bacteria that are in your gut, because neither of these alien neither of these types of alien material inside our body are dangerous. So if you've got a very strong immune response against a food component, that will give you a dietary intolerance. For example, anybody know one? Gluten, yes. Celiac disease. So you have an immune response against gluten protein. Gluten is a protein, so but there might be, there could be some contribution of the gut microbiome to whether you develop an intolerance or not. And it may be to do with the gluten protein itself, or it may be to do with these kind of indirect mechanisms. Okay, so that's kind of intolerance. You don't want to have an immune response against harmless gut microbes, because otherwise you'll be developing, abdominal cramps, diarrhea-like disease, because you're always trying to expel these commensal bacteria. So immune tolerance is very, very important for correct function of the gut, and commensal bacteria play a direct role in educating the gut immune system. So, for example, Bacteroides fragilis. So this is one of these Bacteroidetes. It's a gram-negative anaerobe, frequent in the gut. It secretes this polysaccharide, which has a role in activating regulatory T lymphocytes. And globally, the effect of these lymphocytes is to induce tolerance against this bacterium. Another mechanism, kind of in the same for the same effect, is that several species will produce butyrate as a fermentation product, and in particular Fecalibacterium prosnitzii. So this is one of the gram-positive anaerobes, one of the firmicutes, a major component of the gut microbiome. They'll produce butyrate, and this also has an effect on the developing immune system. So it induces tolerance. So it's kind of telling your immune system, we've got helpful fermentary bacteria here. Don't reject them. And another way that gut bacteria interact with the gut immune system is from these guys, segmented filamentous bacteria, which are very interesting because they grow on the surface of gut epithelial cells. They don't grow in culture at all. They're absolutely symbiotic. It's only been possible to grow them for about 2 years in a co-culture with epithelial cells. And these bacteria are very important for the Th17 response based on the secretion of interleukin 17. And that's very important for the development of gut lymphoid tissue and secretion of IgA. And if you don't have these bacteria in the gut, again, this is studies from mice, then mice are very, very susceptible to other types of infections. Pathogenic bacteria can easily infect the gut, infect the intestine, if these bacteria are not present. So you have commensal bacteria performing both functions, okay? inducing tolerance against harmless bacteria and helping to put in place the immune effector mechanisms that are necessary to resist infection by pathogens. So, on the subject of resisting pathogens, commensal gut bacteria are very important to prevent colonization by pathogens. And this is really a problem in a hospital context when patients are getting treated by antibiotics, for example, somebody who's had an operation. To prevent infections after the operation, they get antibiotics. This will have a severe effect on their gut microbiome. And that will make those people more susceptible to picking up pathogenic bacteria. So one of the big problems in hospitals is Clostridium difficile infections, which can get a foothold because the gut microbiome has been eliminated by the antibiotic treatment. So how do normal commensal bacteria prevent this kind of colonization? One aspect is they're just taking up all the space. Okay, so they're blocking all the ecological niches. So there's no space for pathogens to start growing. And in a more active way, many commensal bacteria produce antimicrobial compounds. So Escherichia coli produce a bunch of proteins called colicins that are toxic for pathogens. So they're actively destroying pathogens that arrive. So knowing this, some people have kind of thought, okay, so we know that when you've got a normal gut flora, you'll be more resistant to colonization with pathogens. For someone who's got a gut infection, is it possible to treat them by putting back a normal gut flora? And the way you do it is this. It's called a fecal transplant. So what you want to do is pick up feces from a healthy person, mix them all up in the blender, put this into tubes, and then seed the gut of the person who's got, for example, Clostridium difficile infection with this preparation of healthy gut flora. So you can do this either with a line that goes in through the nose and start seeding the small intestine, or through the rectum and seed the large intestine. You don't want to do this one, just get a kind of like a bulb and start with a kind of like DIY, because this will only introduce bacteria into the rectum. It won't actually be very helpful. So this is not something to be done at home. It's for hospital context only. Okay. And so here are the results. This is a group of patients who had a nosocomial Clostridium difficile infection. The reference treatment was vancomycin. So only like 30% of the patients were cured by vancomycin treatment. Even worse if they got a bowel lavage. So that's not so great. But look at this. Look at this. This is the fecal transplant here. Just one infusion. They've got 80% of people getting rid of their Clostridium difficile. 95, almost 95% if it's repeated. So this really worked, putting back a normal gut microbiome into a pathological state. It really helped. So last point here about the gut microbiome is, does it have an effect on metabolic diseases like obesity, type 2 diabetes, which are very prevalent and becoming a real health concern in a lot of countries. So the idea is that maybe the gut microbiome is contributing to this. So there are a few papers published in 2005, 2006 by a group in St. Louis who really set the ball rolling on this one. Okay, so some of them in mice. So this study in mice, they got genetically obese mice. I think these mice have a disruption in the gene encoding for leptin, so when they eat, they don't get satiated, so they carry on eating. So these mice have a weight gain. And they studied the microbiome in the littermates that were obese and lean, okay, so thin. And they found that there were fewer bacteroidetes and more firmicutes in the feces of these mice. So that's an association, right? You can't prove causality if you have an association, because it might be these mice are eating more. So maybe that's why their microbiome is different. But does the microbiome cause them to eat more? That's a different question. So they tried to look at this with an experiment where they got the gut bacteria from these mice and they put them into germ-free mice. Then they got the gut bacteria from lean mice and then they put them into another group of germ-free mice. So this way you can control the causality. And they gave both these groups of mice a very rich diet, and they found that in fact they did gain, these mice here, they did gain more weight. So this is an indication, at least in this mouse model, that the gut microbiome is having an effect on weight gain. Okay. So that's in mice. What about in humans? I would say not such clear data. Okay, so I don't think, but there's been a lot of studies to try and replicate this and see to what extent it's really a problem. So one of these studies was published in 2009. Okay, this is very interesting because it's studying twins, right? So they've got 31 pairs of monozygotic twins, 23 pairs of dizygotic twins, and they're studying their gut microbiome. Because with this kind of study, you can eliminate the effect of genetic factors. Because if you're looking at monozygotic twins, the genetic background is identical. So then you might think, okay, if you've got one who is overweight and one is very thin, and you've got the microbiome that's different, then we can really link these two things because you eliminate the genetic variability. I think it kind of failed in that respect because they said that in almost all cases, the kind of weight profile was shared between the two twins in each pair. So they couldn't really delineate things because you've got children being brought up together, so probably they have a very similar microbiome, they're living in the same house and they're getting the same diet. It's very kind of difficult to delineate direct effects. So anyway, what they found was they had a less diverse microbiome in people here who were obese. Globally, they had fewer bacteroidetes and more actinobacteria, but they didn't really have the same kind of profile with the bacteroidetes firmicute ratio. And I think there have been other studies which have replicated this. Okay, so people who have type 2 diabetes, have clinical obesity, they're going to have a less diverse microbiome in their gut. However, oh yeah, that's another thing, yeah. So this was another study. This was also very interesting. So this is 2013. So twins here who are discordant for weight. Okay, they transferred their gut microbiome into germ-free mice. And once again, the microbiome transferred from a human donor who was overweight caused weight gain in the germ-free mice, in the receiver mice. So there's definitely a causal relationship between the gut microbiome and the phenotype. However, not so clear whether it's easy to modify the gut microbiome in a person, and whether this can be leveraged up to get a therapeutic benefit, for example, in treating people with type 2 diabetes, which is a serious chronic disease, and if people could regain insulin sensitivity, then that would really be a huge benefit. But I haven't really seen a massive breakthrough paper that allows new treatments to be based on this concept. But definitely very, very active area of research. Okay, so that's the gut. How about the skin? So there are people who just want to look at the skin, and skin microbiologists have published this paper here. So bacteria on human skin. So here they sampled different sites on the body and classified them into, sebaceous, damp, or very dry areas of the skin. And in this case, you do get different types of microbiota. So, okay, the main bacterial genus on the skin is going to be Staphylococci, and in sebaceous areas, you'll have Propionibacteria. In what they call damp areas of the skin, Corynebacterium again. And on the dry regions, much more mixed, but they say they have Proteobacteria. So the two main ones we're going to think about are Staphylococci and Propionibacteria. So Propionibacteria, facultative anaerobes, are going to be fermentary bacteria. They colonize hair follicles in the sebaceous glands when people grow into children, enter adolescence, because the composition of sweat changes and contains more fatty acids, which can be used by these bacteria as a food source. So there are two consequences of this. One is they produce volatile fermentation products, so people's sweat starts to smell bad. The second one is if you have too many Propionibacteria growing in your hair follicles, this will cause inflammation and acne. So Propionibacterium acnes is a result of this. Staphylococci don't really have any pathology. Yeah. Okay, so here, Staphylococci, the species we're talking about are going to be Staphylococcus epidermidis and Staphylococcus hominis, not Staphylococcus aureus. And Staphylococcus epidermidis, is considered as a harmless commensal bacterium. So recently, people have been looking at whether it plays a role in educating the skin immune system, just like oh no. Is that okay? Just have a bit of light in the room. To see whether they play a similar role as gut bacteria. So in fact, the answer is yes. Okay, so what happens is that soon after birth, well, this is a mouse model of what is happening in the human system. Now, everybody is born sterile, right? And soon after birth, our skin is colonized by bacteria, just the same way as our guts are colonized by bacteria. And one of the first to appear is Staphylococcus epidermidis. So in a mouse model, when they put Staphylococcus epidermidis onto mice, which actually don't normally carry this bacterium, then this provokes regulatory T cells to come in. So once again, you've got the bacterium that is educating the local immune system to tolerate the presence of this bacterium. And they found that this was, the timing was very important. So Staph epidermidis can induce this tolerance if they colonize the skin very soon after birth. This is why the Propionibacterium acnes is causing problems, okay? Because they don't colonize the hair follicles after birth. They colonize them during adolescence. So at this time, you've got a kind of mature immune system in your body, and they'll recognize Propionibacterium as an invader. So that's why you get inflammation caused by these bacteria. Also, similarly to what we saw with the SFB in the gut, so another group has shown that these Staph epidermidis can help the development of this, once again, this interleukin 17, which is very important in resistance against Candida infection. So Staphylococcus epidermidis, that's the good guy on our skin, okay? It's harmless, helps educate the immune system, and its presence will actually help develop the right type of immune response to repel other pathogens. Now, the bad guy on the skin is Staphylococcus aureus. It's associated with atopic dermatitis. Often, if you have a Staphylococcus epidermidis infection, this will make the dermatitis worse. And this seems to be a direct result of one of the toxins expressed by Staphylococcus aureus that interactivates mast cells, part of the immune system that is involved in allergic responses. Now, Staph epidermidis and Staphylococcus aureus are quite antagonistic, and Staphylococcus epidermidis will actually prevent colonization by Staph aureus. So here, this is a paper just published this year, shows that coagulase negative staphylococci, so that's Staph epidermidis and Staph hominis, commensal species, they produce antimicrobials. Some of them are bacteriocins, others are from a different family called lantibiotics, but they're specifically active against Staphylococcus aureus. So once again, you've got the commensal bacteria that have a very active role in preventing colonization of this colonization by pathogens. Okay, last one, and I think we'll probably finish a little bit earlier, is the vaginal microbiome. And this is quite different because it's less diverse than all the other, than the microbiome in the other sites in the body. So in most women, the vaginal microbiome is dominated by lactobacillus. And 5 different types of microbial community have been described in different women. So 4 of them are dominated by lactobacillus. So it's a different species of lactobacillus in each of these 4 communities. And type 4 has few lactobacilli. So type 4 is like this one here, group 4 microbiome. So you have more diversity. Now, a big difference here is that this more diverse microbiome has been associated with more frequent episodes of bacterial vaginosis. So in this case, a more diverse microbiome seems to be not protective and seems to be, I hesitate to say harmful, but it's more of a risk factor than a protective factor. Okay, so lactobacilli are the good guys in this case. What are they doing? Well, the main mechanism is just by fermenting glycogen that is secreted by the vaginal epithelium. So this will give a low pH, which will inhibit the growth of most other bacteria that are going to come in and colonize the site. Now, it is also true that lactobacilli from the vaginal epithelium will secrete bacteriocins and that are active against other bacteria. So this is not the only mechanism, but it's the main one. Yeah, well, I did this quickly last night. I had to, okay, question about the typos in the last couple of slides here. I have to admit this lecture did not exist on Sunday morning. It had to be ready for the French group for Monday morning. So, and then I just did the English version last night. So the summary for the human microbiome is, really the power comes from these next generation sequencing techniques. Oh, one thing I forgot to say about the gut microbiome studies. The idea that a less diverse gut microbiome is going to be associated with metabolic diseases. Together with that is a kind of a genomic study where the people are looking, the researchers are not just looking at the different species involved, but they're looking at the whole of the genomes that are sequenced. So you can have an idea of how this microbial community is going to metabolize carbon. And it seems that the gut microbiota that's associated with higher weight gain is going to be more efficient in extracting calories from the food input. Okay, so there's a kind of a mechanistic explanation of why some gut microbial communities are associated with obesity and type 2 diabetes. And that's because, you know, from these techniques where you sequence the whole genomes of everything that moves and try and figure out what they can do, what they can't do. What you need to know, which bacteria inhabit which niche, okay? What are the main bacterial groups in the gut, on the skin, in the mouth, and the vaginal mucosa? How these bacteria interact between each other, okay? How do they, well, for the bacterial plaque, and this is an example of how the commensal bacteria interact and cooperate, and how do commensals act to exclude pathogens. And in terms of bacterial host interactions, the main thing is regulation of the host immune system, which is very interesting. So that's it. I finished in 1 hour. If you like, we can start up with bacterial pathogens, or if you like, we can leave that to